How does the mass number of an element differ from the atomic number?
The mass number is the sum of the number of protons and neutrons in the nucleus of an element. Using helium as an example, the mass number of helium is 4. As it has 2 protons and 2 neutrons in its nucleus. Since it has only 2 protons, the atomic number of helium is 2. When the atomic number changes, i.e., the number of protons changes, the result is a different element.
What is Darwin's warm pond?
Darwin's idea of a suitable milieu for the origin of life. On Earth was a little pond gently warmed by sunlight. This can be contrasted to the Oparin-Haldane hypothesis of an electrically active methane and carbon dioxide atmosphere for the origin of organic evolution.
What is cloning?
A clone is a group of cells derived from the original cell by fission, one cell dividing into 2 cells. Or by mitosis, cell nucleus division with each chromosome splitting into 2. Cloning perpetuates an existing organism's genetic makeup. Gardeners have been making clones of plants for centuries by taking cuttings of plants to make genetically identical copies. For plants that refuse to grow from cuttings, or for the animal world. Modern scientific techniques have greatly extended the range of cloning. The technique for plants starts with taking a cutting of a plant that best satisfies the criteria for reproductive success, beauty, or some other standard. Since all of the plant's cells contain the genetic information from which the entire plant can be reconstructed, the cutting can be taken from any part of the plant, placed in a culture medium having nutritious chemicals and a growth hormone. The cells in the cutting divide, doubling in size every 6 weeks until the mass of cells produces small white globular points called embryoids. These embryoids develop roots, or shoots, and begin to look like tiny plants. Transplanted into compost, these plants grow into exact copies of the parent plant. The whole process takes 18 months. This process, called tissue culture, has been used to make clones of oil palm, asparagus, pineapples, strawberries, Brussels sprouts, cauliflower, bananas, carnations, ferns, and others. Besides making highly productive copies of the best plant available. This method controls viral diseases that are passed through normal seed generations.
Who developed plant breeding into a modern science?
Luther Burbank, 1849 to 1926, developed plant breeding as a modern science. His breeding techniques included crosses of plant strains native to North America and foreign strains. He obtained seedlings that were then grafted onto fully developed plants for an appraisal of hybrid characteristics. His keen sense of observation allowed him to recognize desirable characteristics, enabling him to select only varieties that would be useful. One of his earliest hybridization successes was the Burbank potato, from which more than 800 new strains and varieties of plants including 113 varieties of plums and prunes were developed. More than 20 of these plums and prunes are still commercially important today.
What is the latest genome to be made public?
In January 2004, the first draft of the honeybee genome was made available.
What is the structure of a ribosome?
A ribosome is composed of two parts known as the large and small subunits. Each of these is a combination of protein and a type of RNA known as rRNA. At the beginning of translation, the two subunits form a structure around the mRNA molecule as the first tRNA. The one matching the first methionine or fmet arrives. The completed ribosome has niches that hold up to 3 tRNAs at a time. Because a cell has so many ribosomes at any one time, rRNA is the most common type of RNA found in cells.
What is Müllerian mimicry?
Fritz Müller, 1821–1897, a German-born zoologist, described a phenomenon in 1878 in which a group of species with the same adaptations against predation was also of similar appearance. This phenomenon is now called Müllerian mimicry. Müllerian mimics include wasps and bees, all of which have similar yellow and black striped patterns that serve as a warning to potential predators.
Which animal viruses are DNA viruses and which are RNA viruses? Why is it difficult to treat viral infections with medications?
Antibiotics are ineffective against viral infections because viruses lack the structures, e.g., a cell wall, with which antibiotics interfere. In general, it is difficult to treat viral infections with medications without affecting the host cell, as viruses use the host cell's machinery during replication. Several antiviral drugs have been developed that are effective against certain viruses.
What ciliate characteristics give the impression they are complex organisms, although they are in fact unicellular?
Ciliates have many specialized organelles within their single cells. These organelles include cilia for locomotion, two types of nuclei, micronuclei and macronuclei. An oral groove to ingest food, food vacuoles for digestion. A contractile vacuole to regulate water balance, and a cytoproc to expel solid waste particles. Species of the genus Paramecium, which are found in fresh water, respond to their environment as if controlled by a nervous system. For example, when they encounter physical barriers, they react by reversing the beating of their cilia in order to proceed in a new direction.
What are bioluminescent bacteria?
Bioluminescence is the production of light, with very little heat, by some organisms. The light-emitting substance, luciferin, in most species is an organic molecule that emits light when it is oxidized by molecular oxygen in the presence of an enzyme, luciferase. The enzyme picks up electrons from flavoproteins in the electron transport chain and then emits some of the electron's energy as a photon of light. Bioluminescence is primarily a marine phenomenon occurring in many regions of oceans or seas. One example is the Milky Sea found in the Indian Ocean, where the sea appears to have a soft, white glow.
What was the distribution of radioactive fallout after the 1986 Chernobyl accident?
On April 25 to 26, 1986, the world's worst nuclear power accident occurred at Chernobyl in the former USSR, now Ukraine. While scientists were testing one of the 4 reactors at the Chernobyl nuclear power plant, located 80 miles 129 kilometers north of Kiev, an unusual chain reaction occurred in the reactor. This subsequently led to explosions and a fireball that blew the heavy steel and concrete lid of the reactor. Radioactive fallout, containing the isotope cesium-137, and nuclear contamination covered an enormous area, including Belarusia, Latvia, Lithuania, the central portion of what was then the Soviet Union, the Scandinavian countries, the Ukraine, Poland, Austria, Czechoslovakia, Germany, Switzerland, Northern Italy, Eastern France, Romania, Bulgaria, Greece, Yugoslavia, the Netherlands, and the United Kingdom. The fallout, extremely uneven because of the shifting wind patterns, extended 1,200 to 1,300 miles, 1,930 to 2,090 kilometers, from the point of the accident. The accident led to the release of roughly 5% of the reactor fuel, or 7 tons of fuel containing 50 to 100 million curies. Estimates of the effects of this fallout range from 28,000 to 100,000 deaths from cancer and genetic defects within the subsequent 50 years. In particular, livestock in high rainfall areas received lethal dosages of radiation.
What is a metabolic pathway?
A metabolic pathway is a series of interconnected reactions that share common mechanisms. Each reaction is dependent on a specific precursor, a chemical, an enzyme, or the transfer of energy. One of the first studies of metabolic pathways was carried out in 1909 by the British physician Archibald Garrod, 1857 to 1936. His study suggested a link between the inability to make a particular enzyme and inherited disease. The disease was alkaptonuria, a condition in which urine darkens upon exposure to air. Due to the presence of the chemical alkapton. Garrod's discovery was one of the first incidents of a physical manifestation being tied to a specific metabolic disorder.
What is a molecular clock?
The molecular clock is a hypothesis that assumes that random mutation in terms of the substitution of one nucleotide for another, occurs at a linear rate. By comparing the number of nucleotide substitutions between two species, scientists could then estimate the amount of time that has elapsed since they shared that sequence with their most recent common ancestor.
How long can bacterial spores remain dormant?
In 1995, scientists revived bacillus spores from the digestive tract of an amber-encased stingless Dominican bee, Proplobia dominicana, which lived from 25 million to 40 million years ago. Previously documented records based on ampoules that Louis Pasteur collected indicated that spores were able to survive for only 70 years.
Does exercise increase the number of muscle cells?
Exercise does not increase the number of muscle cells. Adult animals have a fixed number of skeletal muscle cells. Exercise, however, does enlarge existing skeletal muscle cells.
What are the major differences between monocots and dicots?
The seed leaves, also called cotyledons, are different in monocots and dicots. Monocots have one cotyledon, while dicots have two cotyledons.
What is a karyotype?
A karyotype is a snapshot of the genome. A karyotype can be used to detect extra or missing chromosomes, chromosomal rearrangements, or chromosomal breaks. Any cell that contains a nucleus can be used to make a karyotype. However, white blood cells seem to work best for human karyotypes. After the cells are cultured, they are killed by using a drug that halts mitosis. And the chromosomes are then stained, observed, and a size order chart is produced.
When was the microtome invented?
Although there were cutting engines as early as the 18th century. The first microtome was invented by Wilhelm His, 1831 to 1904, in 1870. Its development allowed scientists to prepare very thin, uniformly sized slices of tissue for examination under a microscope, instead of the imprecise ones previously prepared using a handheld razor or cutting engines.
What is a biomedic?
A biomimetic is a chemical reagent that can perform the function of a biological molecule. An example is a small molecular mimic of erythropoietin, the protein that causes release of red blood cells. Dolly the sheep was the first mammal to be successfully cloned.
What are the uses of chromatography?
Chromatography is used to separate and identify the chemicals in a mixture. It is useful to, 1. Separate and identify the chemicals in a mixture. 2. Check the purity of a chemical product. 3. Identify impurities in a product. And 4. Purify a chemical product, on a laboratory or industrial scale.
What is a bug, biologically speaking?
The biological meaning of the word bug is significantly more restrictive than in common usage. People often refer to all insects as bugs, even using the word to include such organisms as bacteria and viruses, as well as glitches in computer programs. In the strictest biological sense, a bug is a member of the order Hemiptera, also called true bugs. Members of Hemiptera include bedbugs, squash bugs, clinch bugs, stink bugs, and water striders.
What was Alfred Russel Wallace's contribution to phrenology?
Alfred Russel Wallace became interested in a number of subjects outside of natural science, including the suffragette movement and spiritualism. He is also known for his approval of phrenology, which uses the shape of an individual's skull to make predictions about his or her mental functions. Wallace pointed out that while other scientists talked about the connection between the mind and the body, phrenologists had actually attempted to link physical structures, the skull, with mental attributes, thought processes. although phrenology has been largely discounted as a valid scientific endeavor. In Wallace's time it was just one of a number of areas of investigation into the natural world.
What is DNA sequence analysis?
DNA sequence analysis is part of a subfield of genomics known as structural genomics. DNA sequence analysis results in a complete description of a DNA molecule in terms of its order of nitrogen bases: adenine, thymine, guanine, cytosine. All sequencing projects are based on a similar technology: DNA fragmentation with restriction endonucleases. Cloning of the DNA fragments, and overlapping analysis of the fragments to eventually provide a map.
What is agar?
Agar is a polysaccharide extract of red algae that is used as a solidifying material in microbiological media. Agar was developed as a culture media for bacteria by Robert Koch, 1843 to 1910. Koch was interested in the isolation of bacteria in pure culture. Because isolation was difficult in liquid media, he began to study ways in which bacteria could be grown on solid media. After sterile, boiled potatoes proved unsatisfactory, a better alternative was suggested by Fanny E. Hesse, 1850 to 1934, the wife of Walther Hesse, 1846 to 1911, who was one of Koch's assistants. She suggested that agar, which she had used to thicken sauces, jams, and jellies, be used to solidify liquid nutrient broth. Agar is generally inexpensive and, once gelled, does not melt until reaching a temperature of 212 degrees Fahrenheit 100 degrees Celsius. If 1 to 2 g of agar are added to 100 ml of nutrient broth, it produces a solid medium that is not degraded by most bacteria.
What is the difference between active dry yeast and compressed fresh yeast?
Both active dry yeast and compressed fresh yeast are leavening agents. Active dry yeast comprises tiny, dehydrated granules of yeast. Although the granules are alive, the yeast cells are dormant due to their lack of moisture. Because the cells are dormant, dry yeast has a long shelf life. Active dry yeast becomes active when mixed with warm liquid. Compressed fresh yeast is moist and extremely perishable. It must be stored under refrigeration and used within 1 to 2 weeks.
How is the term mole used in chemistry?
A mole, is a fundamental unit of measure. It refers to either the gram atomic weight or the gram molecular weight of a substance. A mole is equal to the quantity of a substance that contains 6.02 x 1023 atoms. Molecules, or formula units. This number is also called Avogadro's number, named after Amadeo Avogadro. 1776 to 1856, who is considered to be one of the founders of physical science.
Who was Mr. Ed?
Mr. Ed was the talking horse and a star of a television show in the 1960s. When he appeared to talk, the horse was actually responding to cues from his trainer. Movement of a small rope running from his halter through his mouth and held by the trainer off camera would cause Ed to move his lips as if he were speaking. In the real world, so far only birds have been able to mimic human speech.
When offered a choice between plastic or paper bags for your groceries, which should you choose?
The answer is neither. Both are environmentally harmful, and the question of which is the more damaging has no clear-cut answer. On one hand, plastic bags degrade slowly in landfills and can harm wildlife if swallowed. And producing them pollutes the environment. On the other hand, producing the brown paper bags used in most supermarkets uses trees and pollutes the air and water. Overall, white or clear polyethylene bags require less energy for manufacture and cause less damage to the environment than do paper bags not made from recycled paper. Instead of having to choose between paper and plastic bags, you can bring your own reusable canvas or string containers to the store. And you can save and reuse any paper or plastic bags you get.
Which part of the wheat plant, Triticum aestivum, is used to make flour?
Wheat is a monocotyledon grass whose fruit, the grain, or kernel, contains one seed. The endosperm and embryo of the wheat plant are surrounded by the pericarp, or fruit wall, and the remains of the seed coat. More than 80% of the volume of the wheat kernel is made up of the starchy endosperm. White flour is made by milling the starchy endosperm. Wheat bran constitutes approximately 14% of the wheat kernel and is found in the covering layers and the outermost layer of the kernel, called the alurone layer. Wheat germ is the embryo of the wheat plant and represents approximately 3% of the wheat kernel. Athoff, there are nearly 24 species of wheat. The most important ones for commercial use are common wheat, Triticum aestivum, sometimes referred to as Triticum vulgar, and durum wheat, Triticum durum. The varieties of common wheat account for 90% of the wheat grown worldwide. Durum wheat accounts for 5 to 7% of the wheat grown, and all other species account for the remainder of the wheat grown.
What roles do enzymes actually play?
Enzymatic reactions can build up or break down specific molecules. The specific molecule an enzyme works on is the substrate. The molecule that results from the reaction is the product.
What are some biological applications of chromatography?
Chromatography has many applications in biology. It is used to separate and identify amino acids, carbohydrates, fatty acids, and other natural substances. Environmental testing laboratories use chromatography to identify trace quantities of contaminants such as PCBs in waste oil and pesticides such as DDT in groundwater. It is also used to test drinking water and test air quality. Pharmaceutical companies use chromatography to prepare quantities of extremely pure materials. The food industry uses chromatography to detect contaminants such as aflatoxin.
What is the heaviest invertebrate?
The giant clam, Tridacna maxima, is the heaviest invertebrate. It may weigh as much as 122 pounds 270 kilograms. The shells of this bivalve may be as long as 5 feet 1.5 meters.
What is camouflage behavior?
Camouflage behavior allows an animal to avoid predation by appearing to be something it is not. Examples of this include the behavior known as freezing. In addition to the opossum, hog-nosed snakes also exhibit this behavior. Freezing may at first seem counterproductive as an anti-predator strategy. But it turns out that some predators, e.g., frogs and wolves, will not attack a non-moving target.
Do pine trees keep their needles forever?
Pine needles occur in groups, called fascicles, of 2 to 5 needles. A few species have only one needle per fascicle, while others have as many as 8. Regardless of the number of needles, a fascicle forms a cylinder of short shoots that are surrounded at their base by small, scale-like leaves that usually fall off after 1 year of growth. The needle-bearing fascicles are also shed a few at a time, usually every 2 to 10 years. So that any pine tree, while appearing evergreen, has a complete change of needles every 5 years or less.
How has the Human Genome Project changed scientists' estimates of the number of genes in humans?
The Human Genome Project has caused a drastic re-evaluation of the probable number of genes in the genome. As a result of the project, estimates have gone from 100,000 genes to somewhere between 30,000 and 40,000.
What is meant by the modern era of genetics?
Mendel's work was really not appreciated until advances in cytology enabled scientists to better study cells. In 1900, Hugo de Vries, 1848–1935, of Holland, Karl Correns, 1864–1933, of Germany, and Erich von Tschirmach, 1871–1962, of Austria examined Mendel's original 1866 paper and repeated the experiments. In the following years chromosomes were discovered as discrete structures within the nucleus of a cell. In 1917 Thomas Hunt Morgan, 1866–1945, a fruit fly geneticist at Columbia University, extended Mendel's findings to the structure and function of chromosomes. This and subsequent findings in the 1950s were the beginning of the modern era of genetics.
What is gene therapy?
Gene therapy involves replacement of an abnormal disease-causing gene with a normal gene. The normal gene is delivered to target cells using a vector, which is usually a virus that has been genetically engineered to carry human DNA. The virus genome is altered to remove disease-causing genes and insert therapeutic genes. Target cells are infected with the virus. The virus then integrates its genetically altered material containing the human gene into the target cell, where it should produce a functional protein product.
How many chloroplasts are in plant cells?
Unicellular algae may only have one large chloroplast, whereas a plant leaf cell may have between 20 and 100.
What three groups are included in the flatworms?
Flatworms belong to the phylum Platyhelminthes. They are flat, elongated, achylomate animals that exhibit bilateral symmetry and have primitive organs. The members of the flatworms are 1. Planar eons, 2. Flukes, and 3. Tapeworms.
What is an allele?
An allele is an alternative form of a gene. There are usually two alleles for each gene, although the number may vary from one trait to another. Each individual inherits one allele from the mother and one from the father. Alleles for a trait are located on corresponding loci on each homologous chromosome.
Where are bryophytes found?
Mosses, liverworts, and hornworts, collectively known as bryophytes, are often found in moist environments. However, there are species that inhabit almost all environments. From hot, dry deserts to the coldest regions of Antarctica. They are most noticeable when they grow in a dense mass.
Which plants most commonly form ectomycorrhizal relationships?
The most common plants associated with ectomycorrhiza are trees and shrubs growing in temperate regions. These trees include pines, firs, oaks, beeches, and willows. These plants tend to be more resistant to extreme temperatures, drought, and other harsh environmental conditions. Some ectomycorrhizal fungi may provide protection from acidic precipitation.
What is a niche?
A niche is similar to the job description of a population within a given species. However, instead of describing how many hours individuals will work, it describes features such as whether the species is active at night or during the day, what it eats, where it lives, and other aspects of day-to-day life. From one environment, or community, to another. The niche may vary depending on how much competition the species faces.
Is there movement among the molecules that comprise a cell membrane?
A cell membrane is mainly composed of phospholipids and proteins, which are two types of bioorganic molecules. Within the membrane, phospholipids are able to move laterally. Depending on temperature and fatty acid composition, phospholipids generally move faster than proteins. The proteins drift about slowly in a body of lipids, much like icebergs in an ocean. Proteins are able to change shape, also known as conformation. For example, carrier proteins are able to bind specific molecules, such as glucose, in order to provide transportation for the molecule. Once glucose is attached to the carrier protein, the protein changes shape and ferries the glucose inside of the cell.
When was the bald eagle adopted as the national bird of the United States?
On June 20, 1782, the citizens of the newly independent United States of America adopted the bald or American eagle as their national emblem. At first the heraldic 268 artists depicted a bird that could have been a member of any of the larger species. But by 1902 the bird portrayed on the seal of the United States of America had assumed its proper white plumage on head and tail. The choice of the bald eagle was not unanimous. Benjamin Franklin, 1706 to 1790, preferred the wild turkey. Oftentimes a tongue-in-cheek humorist, Franklin thought the turkey a wily but brave, intelligent, and prudent bird. He viewed the eagle on the other hand as having a bad moral character and not getting his living honestly, preferring instead to steal fish from hard-working fish hawks. He also found the eagle a coward who readily flees from the irritating attacks of the much smaller kingbird.
What is genetic imprinting?
Genetic imprinting is a form of epigenetic inheritance. It is demonstrated where there is a different expression of an allele depending on the parent that transmits it. An example in humans is that of two medical syndromes, both of which result from a deletion on chromosome 15. In Prader-Willi syndrome, originating from a deletion on the paternal chromosome 15, affected children have small hands and feet, short stature, mental retardation, and are obese. However, in Angelman syndrome, originating from a deletion on the maternal chromosome 15, affected children have a large mouth and tongue, severe mental and motor retardation, and a happy disposition, accompanied by excessive laughter.
What is an ion?
An ion is an atom that is charged by the loss or gain of electrons. For example, when an atom gains one or more electrons, it becomes negatively charged. When an atom loses one or more electrons, it becomes positively charged.
How was it discovered that microorganisms are effective against bacterial infections?
British microbiologist Alexander Fleming, 1881 to 1955. Happened upon the discovery of penicillin's use as an antibacterial agent. In 1928 Fleming was researching staphylococci at St. Mary's Hospital in London. As part of his investigation, he had spread staphylococci on several petri dishes before going on vacation. Upon his return he noticed a green-yellow mold contaminating one of the petri dishes. The staphylococci had failed to grow near the mold. He identified the mold as being of the species Penicillium notatum. Further investigation proved that staphylococci and other gram-positive organisms are killed by P. notatum. It was not until the 1940s that Howard Florey, 1898 to 1968, and Ernst Boris Chain, 1906 to 1979, rediscovered the benefits of penicillin and were able to isolate it for medical use. In 1945 Fleming, Florey, and Chain shared the Nobel Prize in Physiology or Medicine for their work on penicillin.
Why do animals pretend to be hurt?
Among bird watchers, the female killdeer is well known for pretending to be hurt. When a potential predator appears within the vicinity of her nest, the killdeer will adopt a posture of wing dragging, making it appear that she is injured and an easy catch. The female will gradually lead the predator away from her nest, eventually flying off when the predator is at a safe distance from the nest.
Do animal studies predict human behavior?
George Romanes, 1848–1894, was one of the first scientists to investigate the comparative psychology of intelligence. He believed that by studying animal behavior, one could gain insights into human behavior. However, his theory was based on inferences rather than direct observations of comparable behavior.
How are nitrogenous wastes metabolized?
Ammonia, urea, and uric acid are nitrogenous waste products. They are the result of the breakdown of various molecules including nucleic acids and amino acids. Some amino acids are used for new protein synthesis, while others are oxidized to generate energy or converted to fats or carbohydrates that can be stored. Once broken down, the amino group NH2, containing 1 nitrogen and 2 hydrogens, must be removed or the animal will eventually be poisoned. Ammonia is the most toxic of the nitrogenous wastes. It is formed by the addition of a third hydrogen atom to the NH2 group. Both urea and uric acid are less toxic forms of nitrogenous waste but have greater energy requirements for their production.
Why do ecosystems need decomposers?
While energy flows through ecosystems in only one direction, entering at the producer level and exiting as heat and the transfer of energy as biomass, to consumers, chemical compounds can be reused over and over again. In a well-functioning ecosystem, some organisms make their living, their niche, by breaking down structures and recycling the compounds. These organisms are known as decomposers. Without these organisms, the chemicals used to build a tree would remain locked in the tree biomass for eternity instead of being returned to the soil after the tree's death. From this soil will spring new growth, beginning the cycle once again.
What are beta blockers and how do they affect cell function?
In order to understand how beta blockers work, one must understand that most drugs work by binding to receptors on the cell membrane. Drugs called agonists activate cell receptors by causing either an increase or decrease of cell function. Beta blockers are a class of drugs called antagonists. Because they block agonists from binding to cell receptors. An example of a beta blocker is propranolol, which is used to treat high blood pressure and angina. Propranolol works by protecting the heart against sudden surges of stress hormones like adrenaline.
Why does eating chocolate make some people feel good?
Chocolate contains over 300 known chemicals, some of which can alter mood, such as caffeine. Chocolate also contains a small amount of the chemical phenylethylamine, P, a stimulant to the nervous system that makes people feel more alert and gives a sense of overall well-being.
Do cells have specific shapes?
While it doesn't seem that animal cells have a specific shape, they all have a cytoskeleton. The cytoskeleton enables cells to assume and maintain complex shapes, such as the star-like shape of a neuron or the biconcave shape of a red blood cell. Epithelial and connective tissues also have very particular shapes. The columnar epithelium resembles a wall of bricks. Plant cells usually have a specific shape due to their rigid cell wall, which decreases the elasticity of the cell.
What are the major groups of mollusks?
There are four major groups of mollusks: 1. Chitons, 2. Gastropods, which include snails, slugs, and nudibranchs, 3. Bivalves, which include clams, oysters, and mussels, and 4. Cephalopods, which include squids and octopods. Although mollusks vary widely in external appearance, most share the following body plan: 1. A muscular foot, usually used for movement; 2. A visceral mass containing most of the internal organs; and 3. A mantle fold of tissue that drapes over the visceral mass and secretes a shell, in organisms that have a shell.
How are genes controlled?
Genes are controlled by regulatory mechanisms that vary by whether the organism is a prokaryote or a eukaryote. Bacteria, prokaryote, genes can be regulated by DNA-binding proteins that influence the rate of transcription, or by global regulatory mechanisms that refer to an organism's response to specific environmental stimuli such as heat shock. This is particularly important in bacteria. Gene control in eukaryotes depends on a complex set of regulatory elements that turn genes off and on at specific times. Among these regulatory elements are DNA-binding proteins as well as proteins that, in turn, control the activity of the DNA-binding proteins.
How much water does an average sponge circulate during a day?
A sponge that is 4 in, 10 centimeters, tall and 0.4 in, 1 centimeter in diameter pumps about 23 qts, 22.5 L, of water through its body in one day. To obtain enough food to grow by 3 ounces, 100 g, a sponge must filter about 275 gal, 1000 kilograms, of seawater.
What are some beneficial uses of the sclerotia of the fungus Claviceps purpurea?
The sclerotia of Claviceps purpurea are known as the plant disease ergot. Ergot is used pharmaceutically to produce drugs used to induce labor in pregnant women and to control bleeding after childbirth. Ergotamine, an ergot alkaloid, is used to treat migraine headaches.
Why were the 1950s such an important era of DNA research?
In the wake of World War II, when research was primarily driven by wartime need, researchers returned to basic rather than applied research. The 1950s was a period of intense study as scientists attempted to understand the nature of DNA and the gene. The problem was attacked from both biochemical and structural aspects.
What is tropism?
Tropism is the movement of a plant in response to a stimulus. The categories include chemotropism, a response to chemicals by plants in which uncurling of leaves may occur. Gravitropism, formerly called geotropism, a response to gravity in which the plant moves in relation to gravity. Shoots of a plant are negatively geotropic, growing upward, while roots are positively geotropic, growing downward. Hydrotropism, a response to water or moisture in which roots grow toward the water source. Paraheliotropism, a response by the plant leaves to avoid exposure to the sun. Phototropism, a response to light in which the plant may be positively phototropic, moving toward the light source, or negatively phototropic, moving away from the light source. Main axes of shoots are usually positively phototropic, whereas roots are generally insensitive to light. Thermotropism, a response to temperature by plants. thigmotropism or haptotropism, a response to touch by the climbing organs of a plant. For example, the plant's tendrils may curl around a support in a spring-like manner.
What are the different types of soil?
Soil is the weathered outer layer of the Earth's crust. It is a mixture of tiny rock fragments and organic matter. There are three broad categories of soils: clay, sandy, and loam. Clay soils are heavy with the particles sticking close together. Most plants have a hard time absorbing the nutrients in clay soil, and the soil tends to become waterlogged. Clay soils can be good for a few deep-rooted plants, such as mint, peas, and broad beans. Sandy soils are light and have particles that do not stick together. Sandy soil is good for many alpine and arid plants, some herbs such as tarragon and thyme, and vegetables such as onions, carrots, and tomatoes. Loam soils are a well-balanced mix of smaller and larger particles. They provide nutrients to plant roots easily, they drain well, and they also retain water very well. Loams are considered ideal for plant growth.
What are imperfect fungi?
Imperfect fungi are also called deuteromycetes or conidial fungi. They are an assemblage of distinct fungal species that are known to reproduce only asexually. The sexual reproductive features have not been identified and are not used as the basis for classification. In this group, sexual reproduction has not been observed. Most imperfect fungi are thought to be ascomycetes that have lost the ability to reproduce sexually. The best known members of the deuteromycetes are the genera Penicillium and Aspergillus. Whenever a mycologist discovers a sexual stage in one of these fungi, the species is reclassified from the imperfect category to a particular phylum. The phylum selected depends on the type of sexual structures.
What compounds are important in the coloration of flowers?
Color is one of the most conspicuous features of angiosperm flowers. All flower colors are produced by a small number of pigments. Many red, orange, or yellow flowers owe their color to the presence of carotenoid pigments similar to those that occur in leaves. The most important pigments in floral coloration are flavonoids. An important group of flavonoids, the anthocyanins, are major determinants of flower color. The three major anthocyanin pigments are: 1. Pelargonidin, red; 2. Cyanidin, violet; and 3. Delphinidin, blue. Related compounds, known as flavonols, are yellow, ivory, or white. Mixtures of these different pigments, together with changes in cellular pH, produce the entire range of flower color in the angiosperms.
What are the component molecules of DNA?
The full name of DNA is deoxyribonucleic acid, with the nucleic part coming from the location of DNA in the nuclei of eukaryotic cells. DNA is actually a polymer, long strand, of nucleotides. A nucleotide has three component parts: a phosphate group, a 5-carbon sugar, deoxyribose, and a nitrogen base. If you visualize DNA as a ladder, the sides of the ladder are made of the phosphate and deoxyribose molecules, and the rungs are made of two different nitrogen bases. The nitrogen bases are the crucial part of the molecule with regard to genes. Specific sequences of nitrogen bases make up a gene.
How does a cell respond to injury?
In most tissues, injured cells die and are subsequently replaced. However, in the case of nervous tissue, where dead cells cannot be replaced, a number of events can occur. Nerve growth factor is produced by adjoining neurons. This can induce sprouting and growth of previously dormant neurons.
How are animals classified based on what they eat?
Animals are classified based on whether they eat plants, other animals, or a combination of both. Animals that eat only plant matter are called herbivores. From the Latin herba, meaning green crop, and vorus, meaning devouring. Examples of herbivores are cattle, deer, and many aquatic species that eat algae. Animals that eat other animals are called carnivores. From the Latin carne, meaning flesh, and vorus, meaning devouring. Lions, sharks, snakes, and hawks are examples of carnivores. Animals that eat both plants and other animals are called omnivores. From the Latin omnes, meaning all, and vorus, meaning devouring. Humans, crows, and raccoons are examples of omnivores.
How do continuous feeders differ from discontinuous feeders?
Continuous feeders, also known as filter feeders, are aquatic animals that constantly feed by having water filled with food particles, e.g., small plankton or fishes, entering through the mouth. Continuous feeders do not need a storage area, such as a stomach, for food. Discontinuous feeders must hunt for food on a regular basis. They need a storage area to house food until it is digested.
What are the advantages of gel electrophoresis?
In gel electrophoresis, the molecules are forced across a span of gel, usually agarose. Gels are easy to modify and have an excellent power of separation. The frictional force of the gel material acts as a molecular sieve, separating the molecules based on their size.
What are the most common macromolecules used as energy sources by cells?
Cells use a variety of macromolecules as energy sources. Carbohydrates, lipids, and even proteins can be metabolized for energy. ATP and related compounds are also used as temporary energy storage vehicles.
What is a gene?
A gene is a specific sequence of DNA that contains the molecular recipe for a polypeptide. A polypeptide is a subunit of a protein.
Other than DNA, what else can nucleotides be used for?
Nucleotides can also act as messengers or modulators. For example, the nucleotide adenosine, adenine plus ribose without the phosphate, may be the most important type of immunomodulator, a compound that increases or decreases neuronal activity. Adenosine is used clinically to stop the heart when it is beating erratically. The natural heart pacemaker cells then return the heart to its normal rhythm. Adenosine is also thought to play a role in feelings of fatigue or tiredness. Neuroscientists currently theorize that caffeine works to maintain alertness by interfering with the reception of adenosine on the cell surface.
Why is pH so important to life?
The concentration of hydrogen ions in water influences the chemical reactions of other molecules. An increase in the concentration of electrically charged ions interferes with or influences the ability of molecules, specifically proteins, to chemically interact. Most living systems function at an internal pH that is close to 7. But a biologically active molecule has an optimum pH at which it functions. The optimum pH level varies depending on the molecule and where it functions.
What is a flavor savar tomato?
The flavor savar tomato was produced in response to consumer complaints that tomatoes were either too rotten to eat when they arrived at the store or too green. Growers had found that they could treat green tomatoes in the warehouse with ethylene, a gas that causes the tomato skin to turn red. However, the tomato itself stayed hard. In the late 1980s, researchers at Colgene A small biotech company discovered that the enzyme polygalactouronase, PG, controlled rotting in tomatoes. The scientists reversed the DNA sequence of PG. The effect was that tomatoes turned red on the vine, and yet the skin of the tomatoes remained tough enough to withstand the mechanical pickers. However, before the Flavre Savre tomato was introduced to the market, Colgene disclosed to the public how the tomato was bioengineered. This caused a public protest that led to a worldwide movement against genetically modified organisms, GMO.
What feature of liverworts hints to their name?
Liverworts were named during the Middle Ages, when herbalists followed the theoretical approach known as the doctrine of signatures. The core philosophy of this perspective was that if a plant part resembled a part of the human body, it would be useful in treating ailments of that organ or part. The thallus of thalloid liverworts resembles a lobed liver. Therefore, in line with the philosophy presented by the doctrine, the plant was used to treat liver ailments. The word liver was combined with wort, which means herb, to form the name liverwort.
What are giant tube worms?
These worms were discovered near the hydrothermal, hot water, ocean vents in 1977 when the submersible Alvin was exploring the ocean floor of the Galapagos Ridge. Located 1.5 miles 2.4 kilometers below the Pacific Ocean surface and 200 miles 322 kilometers from the Galapagos Islands. Growing to lengths of 5 feet 1.5 meters, Riptia pachyptila Jones. Named after worm expert Meredith Jones of the Smithsonian Museum of Natural History. Lack both mouth and gut and are topped with feathery plumes composed of over 200,000 tiny tentacles. The phenomenal growth of these worms is due to their internal food source symbiotic bacteria. Over 100 billion per ounce of tissue, that live within the worm's troposome tissues. To these troposome tissues, the tube worms transport absorbed oxygen from the water, together with carbon dioxide and hydrogen sulfide. Utilizing this supply, the bacteria in turn produce carbohydrates and proteins that the worms need to thrive.